Hello dear students, welcome to Experimental Techniques in Material Characterization lecture number 30. From these lectures, we will start our discussion on the Raman spectroscopy. So the first part is introduction to Raman spectroscopy. So let's proceed towards the first part. So we will start our discussions from a question that what is Raman spectroscopy? So let's have some formal introductions of the Raman spectroscopy. So Raman spectroscopy is a non-destructive chemical analysis technique which provide detailed information about the chemical structures, phase and polymorphy, crystallinity and molecular interactions. So this kind of the technique is based upon the interaction of light with a chemical bond within materials. So Raman spectroscopy is a light scattering technique whereby a molecule scatters incident light from a high intensity laser source. So most of the scattered light is at the same wavelength as the laser source and does not provide useful information. This is called Rayleigh scatter. So here you can see, we have shown the typical process of the light scattering in the Raman technique. So here you can see that this is a molecule and we have a laser light of particular wavelength. So once we shine this laser light on the molecule, so after that is being scattered. So if the scatter radiations is of equal wavelength as that of the shining laser light, so we call that Rayleigh scattering. And we remember this kind of scattering it does not provide useful information about the material. And the other case is if we have a small amount of the radiations or the light, and it is being scattered at different wavelength, so then we call that Raman scattering. Or what we mean to say, that if we shine a laser light and that laser light it being scattered at a frequency at a wavelength slightly greater than that of the shining laser light, so that we call a Raman scattering. And it has the informations about the material. So this kind of scattering is called the Raman scatter, and it contains information about the sample. So the typical Raman spectrum which we obtain as a result of the Raman spectroscopy is the one here you can see. So this is a typical Raman spectrum of aspirin, and the answered image which you can see it here it's the detail. It shows the detail of the content found in the material. So a Raman spectrums basically features a number of peaks showing the intensity and wavelength positions of the Raman scattered light. So each peak corresponds to a specific molecular bond vibrations, including individual bond such as carbon-carbons, double carbon-carbon bond, nitrogen oxygen bond, and carbon hydrogen bond, etc. And a group of bonds such as benzene ring, breathing mode, polymer chains vibrations, lattice mode, etc. So information provided by the Raman spectroscopy, normally the people ask the questions that what are the kind of informations we can get from the Raman spectroscopy. So remember, Raman spectroscopy probes the chemical structures of materials and provide information about chemical structure and identity, phase and polymorphism, intrinsic stress and strain, contaminations and impurity. So typically a Raman spectrum is a distinct chemical fingerprint for a particular molecules or materials and can be used to very quickly identify the material or distinguish it from the others. So Raman spectrum libraries are often used for identifications of materials based on its Raman spectrums. Libraries containing thousands of spectra are rapidly searched to find a match with the spectrums of the analyte. So Raman is used for macroscopic analysis. So a typical Raman macroscope, you can see it here. Maybe you might have one in your laboratory. So this is it's a typical modern Raman a microscope. I mean that this is linked with the computer screen. So whatever you put here, it's a very simple technique. You just put the sample, you adjust it with the help of optical microscope, and you get the result for that. So how do the thing happens? Let's have some formal discussions that Raman's spectroscopy can be used for macroscopic analysis with special resolutions and the order of 0.5 to 1 macrometer. Such analysis is possible using a Raman microscope. So a Raman microscope couples a Raman spectrometer to a standard optical microscope, which you can see it here. This allow it for high magnification visualization of a sample and Raman analysis with a macroscopic laser spot. So Raman macro analysis is very easy. So how much easy is that? What actually you need to do? You simply place the sample under the microscope, focus it, and make the measurement. You just need to put the sample, you need to focus it with the help of optical microscope, and then you take the measurement. So it's very simple analysis. You don't need a special training for sample analysis where with the help of a Raman microscope. So a true confocal Raman microscope can be used for the analysis of micron sized particles or volume. It can be used for the analysis of different layers and multi-layered sample, for example polymer coating, and of contaminant and feature beneath the surface of a transparent sample, for example impurity within glass and fluid like gas, inclusion and a mineral, etc. So type of a sample that we can analyze with the Raman. So Raman can be used to analyze many different sample. In journal, it is suitable for the analysis of solid, powder, liquid, gel, slurries, and gases. It can be used to analyze inorganic, organic, and biological materials. It can be used to analyze pure chemicals, mixtures, and solutions, metallic oxides, and corrosions. But be remember, the Raman spectroscopy technique and journals are not suitable for the analysis of metals and their alloys. So typical example of where Raman is used today include art and archaeology, that is characterization of pigments, ceramics, and gemstones; carbon materials, that is structures and purity of nanotube, defect or disorder and the materials. In chemistry, it can be used to find out the structures, purity, and reactions monitoring of the synthesized sample. Similarly, it can be used in geology for mineral identifications and distributions, fluid inclusion, and phase transitions. Along with that, Raman spectroscopy is also used in lab sciences for single cell and tissue analysis, drug interactions, and disease diagnosis. So the people also use it in pharmaceutics. So in pharmaceutics, it can be utilized for content uniformity and component distribution inside a particular chemicals or drugs. Semiconductor industry also utilize micro or Raman spectrometers for finding the purity, alloy compositions, and intrinsic stress and strains. Uh, Raman spectrometers, since we mentioned in the beginning that it's a light, high intensity laser light. So one may ask the questions that light has a spectrums that range, that has a particular range, from radio to gamma. So here you can see where actually Raman's, the Raman lies. So here you can see that we have this spectrum. So in these spectrums, you can see here that the Raman lies between the infrared and UV. So means we have NIR close along with that visible and UV. So this is the typical range for the Raman. So that's all we have for this introductory lecture. Thanks for watching, but stay tuned for the next lecture on Raman spectroscopy, because in that we will discuss the Raman effect. For the Raman effect, we will have a detailed discussion of classical and quantum mechanical approaches. So stay tuned with the next lecture. Till then, bye bye.